up, and let's just use slideshow, I think is safer. I'm going to take that off. Okay, very good. So our work really began by looking at the ultimate limit of miniaturization. And I'll show you a couple of very brief examples of that. And in order to be able to do that work, we needed two capabilities. One was a set of microscopes that could record with atomic resolution or better structure, function, and spectra. And those are not the subject of today's talk. We also needed the ability to place the functional species into controlled environments. And really, what we ended up doing was adding the chemical dimension of nanolithography, where we could place a functional molecule into an environment in which we controlled hydrogen bonds down to 1/10 of an angstrom, and at the same time controlled the placement of molecules all the way out to the centimeter scale. And so what happened as we were doing that is that a neuroscience colleague, Professor Ann Andrews, came to us and suggested that since we could gain that chemical control, we could also control and manipulate biological interactions. And so with her, we've developed a series of sensors, initially targeting listening and on chemical communication in the brain. And that was the basis of the Brain Initiative in the United States that she and I helped put together, as well as sensors in other areas such as biomarkers, DNA, RNA, and so forth. I won't have time to talk about that work today, but you might want to have her back to give a talk on it. The next person to come along and see that our work might be useful in an area of interest to him was a postdoc in my laboratory, Steve Jonas. He's now on the pediatrics faculty at UCLA. And what he challenged us to do was to be able to edit the genes in cells at 1 billion cells per hour in a safe way, meaning no viruses, no harsh chemical or physical treatments. And in fact, we came up with 6 different ways to do that. And we had a host of collaborators on the engineering side. But I would say, importantly, we have a series of clinicians who specialize in bone marrow transplants. And so all our work was targeted at getting from where we were in the laboratory into getting these corrected genes into patients. And so we took on diseases, as I'll show you, such as sickle cell and thalassemias, where hemoglobin is miscoded, and also rare immune diseases, and then also cancer immunotherapies. And so the head of the CAR T cell facility in pediatrics at UCLA is Satira de Oliveira, and I'll talk a little bit about that work that we do together. That led to work in diagnosing and tracking the treatment in different cancers and liquid biopsies, and I'll have a chance to talk about our recent work there. This was really inspired and in collaboration with H.R. Tseng in pharmacology here for the circulating tumor cells and exosomes, and with Namjun Cho at Nanyang Technological University for viruses and some of our other work. And again, it's all about, can we do something useful with these structures that we build and devices that we build. And along the way, we also learn a lot of interesting science, and we advance this really 4th dimension of lithography, that chemical dimension. Okay, we work in tissue engineering in the same vein, but we won't have time to talk about that today. Someone can ask me about that, or you can reach me offline if you're interested. Okay, so where did all this come from? As I mentioned, we learned to place individual molecules, pairs of molecules, lines of molecules. Once upon a time, I was the first one to move atoms around with a scanning tunneling microscope before my postdoctoral mentor, Don Eigler, learned to spell out IBM. But there were reasons that we were moving atoms, and at that time was to find out what was underneath them. So we realized that we had this capability in nanotechnology of going all the way down to the very smallest scales in terms of control. And then we also learned to use chemical interactions to advantage to build structures that would be of interest to us. It was really curiosity driven on the one hand, but then also seeing how far down in terms of function we could go in not biological systems at that time, but systems inspired by biology, where we could go from quantum mechanics to mechanical engineering in experiment theory and simulation. So, for instance, we were particularly interested in understanding what we could image with a scanning tunneling microscope, how we could isolate individual molecules and pairs and lines of molecules at defects in these monolayer films. And our early work was in collaboration with my colleague, first at Bell Laboratories and then at Penn State, Dave Valera, who with Ralph Nuzzo at Bell Laboratories developed these self-assembled monolayers of alkane thiols on gold that I think many of you are already familiar with. And then what we learned was that the thinking at the time that the molecules went down randomly and irreversibly and were mobile on the surface once they got there, that all those ideas were wrong, and that the molecules were dynamic, they phase separated, they moved, and we learned to control those interactions and those dynamics to the point where we could systematically eliminate different kinds of defects and control the types of defects that remained to isolate the molecules that we cared about and also to determine their interactions with their surroundings and thus their stability. And so, for instance, in the case of the molecular switches, as I said, we would design hydrogen bonds into the surrounding matrix so that we could stabilize one state or another, and we could design molecules that could or could not show different forms of function. And skipping many years, we developed this idea into the idea of tethering chemical neurotransmitters on surfaces, which had been a goal of Professor Andrews, where we could capture proteins from the brain involved in neurotransmission to do a kind of functionally directed proteomics, but then take on her long-term goal, which was to listen in on chemical communication and understand how a neural circuit functioned and to be able to stimulate it, measure the output, and ultimately to be able to predict that. And that became the Brain Initiative, and we were able to go to the White House for President Obama's announcement. If you're interested in that work, there's a paper from 2018 that describes how we do those measurements. Okay. That led to, and I'm sort of taking you through without going through the the data and science here, to the Microbiome Initiative. There was actually, after the Brain Initiative, another one called the Precision Medicine Initiative that was viewed as a failure, because whereas nanoscientists had drawn together different communities for the Brain Initiative and said, look, we need electrical engineers, we need physicists, we need chemists, we need neuroscientists, we need data scientists, and so forth, the Precision Medicine Initiative group didn't do that. And so the White House asked us to lead the technology roadmap for the Microbiome Initiative as nanoscientists because of the communication skills that we had developed, and because we would draw in whoever we needed, whether it was in oceanography or atmospheric science or soil science or synthetic biology and so forth. And we did that, and we again published the technology roadmap in ACS Nano before the president's announcement of that third and final initiative of the Obama terms. Okay, and then let's get to where we are with Steve Jonas, who I mentioned earlier. So he came to us after his MD, PhD, and residency, and he was in the special position that guaranteed him a faculty position at UCLA when he was done. And so he could really focus on setting up his career and his collaborations. And his idea was to be able to take the hematopoietic stem cells from the bone marrow of patients, and for patients such as those with sickle cell or thalassemias that have hemoglobin miscoded, to correct that in the hematopoietic stem cells while the patients were still in the office, rather than sending the stem cells off to a facility for a few months that's booked up for months or sometimes even years in advance where the cost per dose in that case is $500,000 to $2 million. And so the hope of treating 300,000 patients a year with each of those two diseases is really not possible. And so what we needed to do was come up with an economical, safe way to treat these patients where we didn't give them, as in the viral transfection, the possibility of an off-target cancer, or in the case of the harsh chemical and physical treatments, to disrupt the stem cells. And so, as I mentioned, we came up with half a dozen different ways to do that. I'll show you about 1.5 of those, and these have now been commercialized, and we have a startup company that came out of the laboratory, Transfecta Biosciences, that's trying to get these to patients. The same ideas work with CAR T cells and cancer immunotherapy, but I won't have a chance to talk about that today. So here's the current state of affairs. In a university laboratory setting, the viral transfection might be $100,000 per dose. There's also these fairly harsh chemical treatments that open up the cell membrane, or one can electrocute the cells in order to make holes and get payloads in, but that also damages the stem cells. And there was a recent and inspiring success at MIT from Bob Langer, Dana Anderson, and Klaus Jensen, in which cells were squeezed through constrictions in microfluidic channels, and that opened up transient pores in a way that I'll show you and that we had previously understood. But the problem they've been having is that these channels clog. And so our initial look at this problem was to say, how do we keep channels from clogging? And we came up with a series of ways to do that. In 2 of those, we use omniphobic coatings on the surfaces. In another, we redefine the channels such that they're really only held together with sound waves. And so there's nothing to clog in that case. And as I'll show you, there are 2 different ways to run the acoustofluidics, and we'll talk about that one in a little more detail, because I think it's the most promising on the one hand, and it's also the furthest along towards our goal. But in fact, in all these cases, we're able to get 12 million cells per hour processed per channel. So by going up to 100 channels, we've already been able to reach the goals that we set at the start. Okay, and then, I'll add, and we're going to talk about this a little bit later, that on the other side of this is monitoring diseases. And so we can use some of the same strategies, as you'll see, and some of our inspiration came from the capture of circulating tumor cells done by H.R. Tsang, our colleague in pharmacology. And we learned something from what he was doing. We'll come back to that later in the talk. Okay, now let me back up. So almost 20 years ago now, we had figured out how to get molecules to go where we wanted them on flat surfaces. And so we wanted to look for a bigger challenge. And so we said, well, what are other important surfaces and interfaces? How about cell membranes? Could we get molecules to go where we want them to? Could we establish domain boundaries, which were so important in our control on flat surfaces? And we realized that, number one, we needed different strategies for control. And number two, we needed different techniques to measure them. And so we started out with model systems, these giant unilamellar vesicles, and we used fluorescence probes to say where the different molecules were within the membrane. And the simplest way that we could think to control the placement of the molecules was by curvature. And we showed in our early data that indeed we got phase separation based on curvature, and we could do the measurement with fluorescence. And we published the first work in Journal of Biological Physics, and basically nobody read it. And a little while later, Watt Webb recorded much, much more beautiful and controlled data and published that on the cover of Nature. And that kind of thing kept happening to us where, I thought we had the right idea and we were making headway, but it became apparent that other groups were going to be able to do and would do what we were planning. And I always feel like we should do something in our laboratory that no one else can do. And so we eventually dropped these experiments before we moved to UCLA in 2009. But let me show you two of them along the way from which we learned about control that has played into our work in cellular therapies. And along the way, we also convinced some of our colleagues, particularly Andy Ewing, who's now at Chalmers, and Nick Winograd at Penn State, to study this placement of molecules also. And so they showed in these tetrahymena that when the cells come together and form these junctions through imaging mass spectrometry, that there were different molecules in those places than there were in other parts of the cells. We learned to control shape in a number of different ways. We could use optical trapping of a bead inside the vesicle. We could use micropipette aspiration, or my favorite was growing tubulin into microtubules inside the vesicle, such that we had very high curvature at the points, very high curvature along one dimension, and zero curvature along another. In these parts that look like Saturn's rings from end on, and then negative curvature at this junction and normal curvature in the middle, all in the same system. And we could figure out, for instance, how to trigger the degeneration of the microtubules and reproduce the morphology that was found in dying neurons without the proteins that were thought to be necessary. Okay, then the last experiment we did in this series was the following. So Susan Gilmore, who is a postdoc in the laboratory and is now at NIH, wanted to understand what happened when red blood cells were deformed as they went through capillaries. So she was able to recapitulate the cup shape of the cells, and then what she did was stress those, and to our surprise, what we found out was when they were stressed, transient pores opened up in them, and one could get interactions between the inside and the outside of these model cells, and we're able to figure out how big the pores were and how long they lasted. And we had no idea what to do with that, but we thought that hematologists would. And I'm a strong believer in publishing papers in the right journal. So 3 times we sent this paper to the flagship hematology journal Blood, and 3 times they rejected it without review. So we published it in Journal of Physical Chemistry B, and again, not many people read it. But along came this work from MIT in which cells were squeezed through constrictions, and that opened up pores in order to deliver payloads. And we said, aha, that's what you do with it. And so we brought back our knowledge from the membrane days, and then we brought in our chemical functionalization capabilities, for instance, to coat the channels of the membrane, of the constriction, so that the channels wouldn't clog. And we've been able to do that with, for instance, Joanna Eisenberg and her slippery liquid porous infused pore surfaces, SLIPS coatings with lipids fused from biocells with Namjoon Cho at Nanyang Technological University, and importantly with acoustics with Tony Jun Huang, who's a UCLA alumnus who I had mentored when he joined the faculty at Penn State, and he's now at Duke. And so for each project, we would reach out to the expert and pioneering engineer so that we could make the most headway and go on the steepest gradient to get into the clinic. Okay, so back to this. Now for sickle cell, there's just a single gene mutation that miscodes hemoglobin, and when there are hypoxic conditions, then the red blood cells form this characteristic sickle, different shape. And in the developed world, that reduces the life expectancy of patients down to about 40, and they need blood transfusions regularly. And that also causes problems and associated infections. In the developing world, life expectancy is more like 10 years old. And so with replacing only 10 to 20% of the bone marrow with a correctly coded gene, that would be a cure and eliminate the need for further transfusions. And as I said at the start, we need about 1 billion cells for a 12 kilogram child. And so we said, well, let's figure out how to do that in an hour safely while a patient is in the office, so that we could collect the cells, correct them, and then it turns out if you just reintroduce the corrected stem cells intravenously, they'll go back and nest in the bone marrow. And you don't have to kill off all the bone marrow like in a leukemia patient. You just need 10 or 20% that you ablate with chemotherapy, for instance. Okay, now let me bring you back to when we came to UCLA and what our colleague H.R. Tsang had done. So he had prepared what he called these nano Velcro surfaces. They're sharp etched silicon that can capture different cells, and particularly circulating tumor cells are sticky. And so he used that capability for capturing the circulating tumor cells, but he realized that there was penetration of these sharp barbed wire-like features into the cells. So he said maybe he could use that. And so he developed these payloads that are these supramolecular complexes. He uses host-guest chemistry, and they can get rather large payloads, larger than what one can get with viral transfection, that would include, for instance, CRISPR-Cas9, either protein machinery or coding for it, the guide RNA, and some of the pixie dust to help get into the nucleus. And so we already had these payloads ready to go. He showed that he could load these payloads onto the sharp features and get them into the cells, but just like a human jumping onto barbed wire, it's easier to get on than it is to get off. And so he wasn't able to use those cells further. He could show that they were transfected, but because they're really stuck on the barbed wire, they would be shredded if one used conditions to try to remove them. And so we first started out saying, well, we can come up with sharp, smooth spikes. And so just like using a sharp knife to cut, it does less damage. We were able to make something like he had. Back to the channels for a moment. Here's in the case of the microfluidic channels from MIT, you can see how those get clogged up. And that happens on the order of thousands of cells, and so maybe 1 million times too few cells to do what we said we needed. And so one of the first things we did is we used the coating, the omniphobic coating that Joanna had developed, and she had a startup company that Phil Kim was formerly the chief scientist for. It used to be called SLIPS, and now I think it's called Applied Surface Technologies. And really it took inspiration from the pitcher plant in the jungle, which when it rains, the water is soaked into this porous lip such that insects and even small rodents like mice can't hold on, fall in, and are eaten by these carnivorous plants. Well, Steve Jonas is a pediatrician, and he didn't want to tell kids, about carnivorous plants. So he said, well, it's really like a slip and slide for cells. And what we found is that in the constrictions that we use, we can run, as I said, 12 million cells per channel per hour, and we cannot get the channels to clog when we use the right constriction. Another way that we've taken on to do this, and I think the most promising one, because there are really no additives that we have to worry about the cells picking up, is using acoustics. And this was a method pioneered by Tony Huang, called acoustofluidics, in which either individual cells can be run down the center of a channel and deformed, or large bunches can be used and constrained to the middle. And again, our sweet spot seems to be about 12 million cells per channel per hour. The nice part of these is that we can change the waveforms and the little speaker that we're using basically in order to control what the cells are doing. And we don't need to build a completely new channel if we change, for instance, cell size or type. And there are two different modes to run this. We can either go down the middle of the channel, or we can bounce the cells off the walls And what we found to be most effective is this bouncing method, where electrostatically we attach the payloads to the walls. We thought we would need to first load the payloads and then run the cells and then reload the payload complexes, but we found that we can just run both together and it works perfectly fine. And we get very, very high viabilities, over 90% for the cells, and depending upon the cell type, between about 10 and 40% efficiency of transfection, which is already competitive. Because we can cycle with that high viability the cells for more than one treatment, we think we're going to be able to get to quite high efficiencies this way. And one other important thing to do is to make sure that we get into the nucleus. And so we collaborate with the leading person at UCLA in terms of nuclear penetration, Steve Young. And so each time we do these measurements, we confirm that we've actually gotten the systems all the way into the nucleus of the cells. We've done this for peripheral blood mononuclear cells, and we've also done this for human hematopoietic stem cells. Each one of these experiments is about $5,000 worth of cells. So this was just our very first experiment. We're mostly optimizing on model cells at this point. You can see, even in that first step, we got these very high viabilities and quite high efficiencies of the stem cells that we need in order to treat these patients. We do comparably in the CAR T cell work. So now for the remainder of the talk, let me focus on the liquid biopsies, in which we capture from the blood very rare cells that can be attributed to tumors, or even vesicles that come from those tumors. For the cells, we're able to do individual cell analyses, and in that way we can look at the heterogeneity of the tumors. In the case of vesicles, we're not there yet in terms of single vesicles, but the technology is developing to enable that to happen, and we should expect that to come before too long. So again, I show you a little bit of the early work of H.R. Tseng. We've advanced that further. I won't have a chance to talk about viruses and capturing them, but in that we work with Namjoon Cho. And then we've recently been focusing on a pediatric bone cancer called Ewing Sarcoma. And in that case, the leading clinician in California is Noah Fetterman, who's on our pediatrics faculty. We also work with the head of mechanical engineering for reasons that'll become clear in a moment, Tim Fisher. Okay. As I mentioned, these cells and vesicles come from tumors. They could be responsible for metastasis, and the particular cancer we're focusing on is highly metastatic and not only produces large numbers of circulating tumor cells relatively, but those are heterogeneous, and there are interesting things to learn about them. And we know quite a bit about what's on their surfaces, unlike other cancers. And so we can develop this technology for Ewing sarcoma, and later when we learn more about other cancers, apply it there too. And so in this particular case, we're looking at non-small cell lung cancer, circulating tumor cells. And what we did is we used an antibody that would selectively capture the circulating tumor cells. And into our surface functionalization, we built a breakable link. And so once we capture these circulating tumor cells, when we want to let go, we just put in this molecule dithiotheriitol, and that breaks the link, releases the cell for further analysis. And as I mentioned, we're able to analyze each one of the cells individually. And so we're able to do this first in terms of a diagnostic, and because these are real patients in our hospital who have agreed to be part of our study, they get the gold standard pathology done as part of their treatment, and then we're able to compare directly with the results that we get. And we've been doing very well on that, and I'll point you to some of these recent papers in these click chips, in which we can capture the cells and then later release them. Well, you don't want to use these sharp features when you're working with exosomes. We'll get to that in just a moment. But I'll point out that we can also use all the imaging and diagnostics that exist within the hospital that the patients are getting in any case to track how we're doing. And so there's a real advantage here in working directly with the clinicians who are treating the patients, and while they're anonymized when we get our information, for instance, the chemotherapy that the patients are getting tracks with what we're measuring. And since we can look at how the circulating tumor cells change over time, we can say, okay, this chemotherapy is effective on this particular aspect of the tumor, but not on, maybe not on some other part. Okay, as I said, if we want to capture vesicles, the exosomes, then we can't use sharp features. And so what we've done is taken inspiration from biology. If I can get rid of that, hold on. Okay, and the intestines have these, these villi, and so what we've done is build that non-sharp features, but kind of a forest, and we can functionalize the sides in the same way, and once again capture the vesicles. In this case, we can't release and analyze a single vesicle, but we can do that in bulk. Okay, let's get on to Ewing sarcoma. So these are cancers that are in mostly young men. Some of them are very athletic and they're highly metastatic. So we do find these cells. There's actually a drug in clinical trials, and our collaborator at Georgetown was responsible for that drug. NOAA gets about 1/4 of the patients in America, and our collaborator Jeff Turetsky gets about another 1/4. So we have a large cohort of patients with which to work. And we know, as I said, quite a bit about what's on the surface of the cells and exosomes. And so we can use these novel biomarkers in the same way. We can use that same release strategy in order to let go of the circulating tumor cells or the vesicles, and we can watch how effective the chemotherapies are as we follow along the patient's trajectory. Now, there's something more that we decided we could do, and this is the very last part of the talk, then I'll leave some time for questions. And that was where we made sharp features that had additional functionality. And so we developed microfluidics in which we could grow these gold nanostars in the channels. We can functionalize those because we know how to do the chemistry of the gold surface to do the selective capture. And then we could use the sharp tips of the stars to give a plasmonic enhancement of the Raman spectroscopy that we could use to show that the cell was captured selectively in the way that we were probing. And that's the work of Yael Vinicombe-Wilson, who's a terrific graduate student in the group, who's actually over in Spain right now, visiting a laboratory there, and postdoc Nai-Hao Chang, who just accepted a faculty position at the University of Houston. And so after we probe and decide, yes, this particular cell is the kind we want, and it's been selectively captured, we can again use that plasmonic feature to gently heat the tip and release the cell. And so this is where our mechanical engineering colleague comes in. Because we're able to measure the temperature locally, we can release just that one cell for further analysis, and we can ensure that we have not destroyed it. And there are different levels of heat that a cell can tolerate. And so we warm it just enough for release, but not enough to damage the cell. And so now what we've been doing is doing the selective capture of the circulating tumor cells from Ewing sarcoma, the selective release, and then we're on to the single cell analyses for the patients that we were targeting. So I'm particularly excited about the prospects for this this device and this method, because we get the analysis in situ, and we can do an efficient then capture and release. Okay, so I think there's quite a long way to take this. We benefit very greatly from working very closely with the clinicians, knowing what targets to use to go after on the circulating tumor cells and the exosomes. As more biomarkers are discovered, then we'll apply those as well, and we can do that selectively along our microfluidic channels, so we can say, oh, we captured this cell because this biomarker was on the surface. Here's my group, and I tried to put everyone's name on every slide as we went along. Our collaborators around the world, and the people who paid for things in cellular therapies. Really, when we had this idea, we had no preliminary data, and it was these foundations that stepped up and believed in us: Alex's Lemonade Stand Foundation, Tower Cancer Research Foundation, Hyundai Hope on Wheels, and St. Baldrick's, that got us going. Later on, we were able to get money for commercialization, and Steve won this NIH Director's Award that gets his new and independent lab started on this project. And then in liquid biopsies, a little younger project in our group, we've also had help from foundations and now from the NIH. And so, I would say what we learned from taking on problems like this, you saw it came all the way from, it was basically low temperature physics to these problems in biology and medicine. I think, what you want to do is look for a problem that makes you want to jump out of bed in the morning. And once you can do that, figure out, who to work with, who has common interests and complementary skills, learn to talk to each other. And that's a real strength of nanoscience. So, in the audience, if you're a student or postdoc in nanoscience, know that you're being trained very differently than people in other fields. And then, add people you need. And, if people aren't able to contribute to one of your projects, they may come into others. And when we train our students to think about what's the most important thing they could be doing on the day, week, month, year, decade, lifetime timescales, then we're really going to make headway on interesting problems, and they should be the problems that that you care about. And so with that, I will stop the sharing and be happy to take questions.
Okay. Yeah, we thank Professor Weiss for this wonderful talk. It's very inspiring. I think I particularly agree with the nanoscience. We are training quite differently with other field. Well, our thinking is different. Our training and working are all different.
Yes.
So yeah, it's quite interesting. So while our staff is gathering some questions from the broadcast, I think I will take the opportunity to ask some questions. I think I have some general questions. I don't work in the biomedicine field, so it's a bit fast for me. But I think one particular thing is I'm quite amazed by these organizers of the works from different fields, quite a broad spectrum of peoples. So I think that's the real strength of nanoscience or even material science in a general way. So how do you actually make such thinking? How do you think about these? You don't even know the things exist. So how do you organize these teams to work this project?
Right. Well, what I would say one key part is I'm curious, and so if under normal circumstances I go to 2, 3, or 4 talks a day, and so I look for things that'll be interesting, and then go and see if there's some way we can contribute. One of my fantasies in moving to UCLA, where science, engineering, and medicine are all around one courtyard, was that people would bring their problems to me, and we would come up with solutions together. And in fact, it worked better than even in my fantasies. And so, once we have this real hierarchy in medicine, the transplant people are on top. Once the transplant people started coming to the office and asking for help, I knew we had won. But sometimes, problems aren't even that hard. You just need some capability or skill. And there are tremendous opportunities for us. But I really did not have training in biology. I took a pass-fail as a first-year undergraduate, and then I spent a sabbatical year in the medical school at University of Washington with the guy who invented the DNA and protein sequencer and synthesizer. but I was, very much playing catch up. And so I depend upon our collaborators to teach me and to teach my students and postdocs. And in my own group, it's really a collection of people from many different fields: chemistry, biochemistry, physics, material science, math, electrical engineering, bioengineering, infectious disease, hematology, oncology, mechanical engineering. And we all have to be able to talk to each other. That's part of the training. And the very first breakthrough, I would say, in the gene editing came from a mechanical engineering postdoc who's now Professor at Tongji University, Xiaobin Xu, who said, "Oh, wait, I have a way to make sharp needles, and instead of the barbed wire, we can have something that, slides the cells back off." And so once you get people thinking that way, that they can contribute, I think it's very addictive. And then you start looking for, what would I like? What am I interested in? What would I like to do? It turns out to be kind of fun to work with pediatricians because they tend to be really nice. And so, at the beginning of our meetings, for instance, in the Ewing Sarcoma group, we don't get to know who the patients are, but, the clinicians are sort of trading notes on how's this patient doing, how's that patient doing. It makes it very personal. And it's very personal to them. If only I had this capability, I could save this kid. And then there are a lot of those children coming through. And so it really motivates the group. Then our medical students who are in training, what we ask them to do is when they go around and learn, they're also doing reconnaissance for us. So they're saying, where's something that doesn't have a treatment? Or what if I had this capability? What could I do? And when we realized for nanoscience, one of the things is the eye is very accessible. So what can we do in the eye where diseases that haven't been treated, where we could have access? And that's been a very productive part of the group. And then, in fact, right after the talk, the head of our eye institute is coming over. It's Friday evening here. So in 45 minutes, he'll come over for a glass of wine and we'll talk about our research and our weeks and our children. And so, it's also kind of binding, once you see that once you show people you have a capability they can use, or you have a brain they can use, or even, the people they should talk to, they'll keep coming back. And it's really interesting for us. And one of my great pleasures is, putting fields together and putting people together. That's what we did at the journal, and that's what we did at our Nanoscience Institute. And that's what I'm trying to do, more globally in the world now, be the matchmaker and sometimes play a role myself as well.
Yeah, thank you. It's very inspiring. I think it's question and answers. These are important for you to for nanoscientists to think. Okay, so we actually have a question from the website. So
great.
One is actually very particular. He asked, what is your current progress of this 1 billion cells in 1 hour?
We can do it.
It seems quite impossible task.
So
It's already solved?
We solved it. We've made multiplex with 10 channels. And in that we do, the limit right now is, can we afford the cells to do the full test?
I see.
And so,
okay, so multiple channels.
We can do 10 channels with a single acoustic source, for instance, at 12 million cells per channel per hour. And so just expanding out to 100, which is a trivial extension, gets us all the way we need to go. And that's why the company, is ready to launch. I think it's looking for financing now if someone's out there listening.
Okay, I think the audience get it. Yeah, they actually managed to do it.
Yes.
So there's another technical question about the silicon nanowires. So why would you need the sharp features to capture the cells? Would a flat face also works the same way?
Flat face does not work as well and is less selective. So the circulating tumor cells are kind of rough, have a kind of rough surface. And so that was the thing, that was HR's thinking, was if you make something like Velcro, right, then it'll capture the circulating tumor cells better, and that seems to work. You also I didn't show the microfluidic systems, but you want to make sure to bounce the cells into the capture surface. If they just flow over the top of it and never sample the surface, that doesn't do any good. And so it's also important to have the, kind of turbulent flow or another flow that'll bounce the cells into the capture surface. And so that roughness then helps there too. In the acoustic fluidics, there's no roughness. We don't use anything sharp there. It's just a wall with an electrostatic attachment for the payloads.
Okay, we do have a question about the acoustics. The question is rather general. So how do someone come with the idea of use the acoustic wave?
That was Tony Wong. Yeah, Tony is really a mechanical engineer by training. Yeah, and he had developed that not with the idea of deforming the cells, but just of controlling objects and then controlling cells in microfluidics. And so that was during his time when his career was getting started at Penn State. And then I think as he moved to Duke, medicine was closer at hand. And then he was ready to start, looking at what he could do with cells. And then we had the expertise, thankfully, to be able to, put it all together. So yeah, when it first worked, we called him up, and he's a good friend. And we sent 4 students over to Duke and made it work within a week, and then brought it back and made it work. And then the students still talk back and forth. Several of them have graduated already, but that just means it's a growing family.
Thank you. So due to the limit, we have one last question. This is about the actual technical issue of the functionalization. So for these channels, are they mostly PDMS, or how do you actually function these inside of the channels, membranes? Or?
Yeah, so the answer is it depends. And different systems will use glass or PDMS or other structures. I've actually been pushing for sapphire because it's such a good thermal conductor. And that's what, in a grocery store, when you have a scanner that reads the QR code or barcode on what you're buying, those are artificial sapphire, and so you can fab them quite easily out of any of those different parts. It just depends what you're going to do with them. And so for the slips coatings that have the lubricants, right, PDMS can be difficult to seal up. Right, it's one thing to make it, if you make something not sticky, then it's not sticky. So, in different cases, we use different materials. All those are in the papers we published. We always say very precisely, what it is we're using in each case.
Okay. I think we are approaching the end. Yes. So I think we should thank you, Prof. Weiss, for this wonderful, inspiring talks and all these interdisciplinary works and medicines. I think more people should jump into these more broad teams and expand your scope of your research. Otherwise, you're focusing on one small area and you're not solving real issues. I think so. Real issues is very inspiring, and you have the emotions and the internal compulsion of you to do these works. I feel really satisfying.
Well, the material, we make the materials smart. I thought that fit your journal well.
Yeah. So we also hope you have to submit paper to our journals in the future in the times. Yeah. Thank you again.
Thank you so much.
Yeah. Hi, Paul.
Hey, there you are. Very good.
Okay. Very wonderful talk. Thanks again to our webinar. Yeah. Thank you.
Thank you. Yeah. Sorry for the trouble with Move. It decided I was a baby on my new computer, and it locked me out of Move. I don't know how to fix that, but maybe someone can investigate it in case I have to use it again. How to change the birthday. I cleared the cache out of my browser and everything, and it didn't work. Luckily, I have a backup computer sitting, because it's happened sometimes. I had a backup computer ready to go, so I got Move loaded on that. And
okay, it's okay. Very wonderful talk. Yes. Again, learn a lot from your talk. Yeah. Thank you. Good evening.
Yeah. Thank you.
Okay.
Great. Thanks for having me. Look forward to seeing you in person soon.
Yeah, I actually work in CNSI when I was in the Prof. Duan's groups. We met actually. Probably you don't remember me. I remember times in CNSI. I always saw you there. Yeah, thank you for the talk. It works out fine, I guess.
Thank you.
Yeah, thank you.
Thank you.
Yeah,
thank you.
Good evening. Yeah, see you.